So thank you very much for inviting me, postdocs. I've had amazing meetings with probably 2/3 of the people in here over the course of the past day and a half, and I have thoroughly enjoyed hearing about all of the cool science. I love coming and giving seminars because I get to hear about this like cross-section of cool stuff that's, I don't usually think about. So thank you all for that gift to me. I need to start by just bearing my reality here, which is I am deeply an empiricist. So if you're wondering what corner I'm coming from the empirical corner, and I will show you that today. And I also am deeply interested in the biology. So I'm motivated by understanding how will biology change in future Earth, right? So what will it look like? Where will we be? And what kind of processes will we see at that point? And so I don't feel like any one tool will get me there. So while I say that I'm deeply an empiricist, I'm also really motivated by hypotheses that are generated from external to my system. So hypotheses that in some cases come from ecological theory. So I will start in my empirical corner, actually just setting the stage for why I think what we're doing in our field, not just me and my collaborators, but broadly in the field, is a really exciting time. So I want to start here, in this iconic moment of 1969, which is an awesome year for many reasons. Yeah, you're calculating how old I am now, right? So this was iconic for a lot of reasons, right? This was the very first image that people had ever seen of Earth in one view, right? So 1969, there were people around the world that were not only looking at this in newspapers, watching it on television, but gathering around the radio and listening to word pictures, right? That were being told about this image of Earth in one view. And those words were really compelling to me in thinking about where we are in ecology now. Those words were tiny, alone, fragile, right? If you look back at these newspaper articles, these are the words that were used to describe Earth. It's not a super weird coincidence that the first Earth Day was just 2 years later, 2 years after this, right? There was this explosion of awareness of the value of Earth, and also that Earth was one thing. It wasn't a local place. So I'm going to pan back before that. This is from about 1900. It's a postcard from the Pacific Northwest. So this is celebrating the infinite fisheries in the Pacific Northwest. What's that? Oh, celebrating the infinite fisheries in the Pacific Northwest. And you look at the size of those fish, holy moly, right? We could never eat through that resource, right? There are so many salmon, and they're so big, and look, we will always be able to feed ourselves. It's about 1900. That was approximately the year that my grandfather was born, right? So we're only talking a few long, albeit, but a few generations there for humans. It was a time when, from my grandfather's perspective, you could always go further, and there was always more space. We could make more food. There were more resources. It was infinite, right? So it's this infinite world perspective. But then there's this iconic image, which is broadly a sort of cartoon of data, but time on the x-axis and population in millions on the y-axis. And I put in a few really key moments for human history in here, ways, technological changes that allowed us to feed a whole lot more people and support a lot more people. Now, let's look at when humans land on the moon. 3.6 billion people on Earth. When my grandfather was born, 1902, but let's say 1900, do you know how many people were on Earth, approximately, in 1900? Give it a guess. 1.2, 800 people on Earth. I'm sorry. No, so about 1.6. You're a little low, but generally in the right range. So about 1.6 billion. So from my grandfather's birth to mine, we had more than doubled. Where are we now? 7. Yeah, way-ish, like more than 7.5 billion people on Earth. And just for you guys, I put in the internet there just to let you know that that was pretty recent and that it hasn't always been around. Okay, so now let's pan forward in time. Since that time, we have figured out through ingenuity and technology and all kinds of incredibly clever ways how to fish all the fish out of the ocean, more or less. Right? We can feed a lot of people, but we're now seeing the result of that, which is deaths are greater than births, and we're seeing a decline not only in abundance, but size. Okay? We're doing the same thing on land. This is one agricultural field in Brazil. That's alarming, right? Brazil, that's rainforest, right? So this region had been rainforest and is now being turned into one very large soybean field. Okay, now let's step back and look at this in aggregate at the scale of Earth. So this is croplands on top, pasture and rangeland below. You can see the extent to which these choices at the scale of a field aggregate up to have enormous impacts around the globe. Now look at the extent of grasslands, and this is a very generous definition of where are grasslands on Earth, including some shrubland and area tundra, things like that. So broadly writ, where are grasslands on Earth? If you visually slide these two maps together, you more than create most of that extent of grasslands. So we have converted virtually all of these grasslands, which is where our food comes from, cows and corn, more or less, converted these grasslands into food production already. Okay, now I'm going to pan back. I'm very intentionally going from global scale to organismal scale and back again, because I want to get at that in just a minute. So these choices at the scale of a field, of a farm, where we're converting areas that had been native grazers into areas that are now dominated by domestic grazers. So we're taking this wide variation in the type of herbivores on Earth and compressing it down to cows and sheep and goats and yaks and just a handful of different species that have fairly similar types of feeding across the world at this point, and displacing those native grazers. Okay, fertilizer production since 1950, right? That's really not that long ago. That has increased by more than 400%, right? Haber-Bosch process is allowing us to use the same area of land and feed more people. It's amazing, but now going back to the scale of the planet, these decisions being made again and again and again at relatively local scales are playing out in global ways, right? So this is a paper by Galloway et al. From 2004, making projections into the future of what will nitrogen that's dissolved in water, where will it come down on Earth? So it looks like this. So it's massively increasing. So that nitrogen deposition is increasing hugely. So this isn't just one thing that's changing. There are a lot of factors that are changing at the same time. And often in ecology, we tackle one factor at a time, right? Because it's a lot easier, right? It's mathematically hard, or it's logistically difficult in the field to tackle a lot of different factors at the same time. But really, this is a multifactorial problem. And if we want to make predictions into the future, we need to understand if these multiple changes interact with one another. And so what will our future look like under changes to nutrients and changes to herbivores that are happening individually at local scales, but aggregate affecting the Earth? And so what if we try to articulate those into questions? So asking, for example, under what conditions do grazers or fertilization control the production or the diversity or the composition of plants? What about multiple nutrients, right? We're not just changing nitrogen. I did talk just about that, but we're changing where phosphorus is, where potassium is, where micronutrients are coming into our ecosystems. And so could these have synergistic impacts, for example, on ecosystems? All right, so as an empiricist, now I'm going to step back into my empirical corner, which is where I go when I try to think about a problem. I'm like, okay, how would this play out in my field? What would this look like? Okay, so this is a field at Cedar Creek. I'm standing up on the hill. Anyway, right, it's not much of a hill. But I'm looking down over my field and I'm saying, okay, I've done this experiment at my site. So at my site, I've excluded herbivores, I've added fertilizer, and I can look at it and I can say, oh, wow, that's really amazing. Look at that plot. You can see the greenness here is a lot, it's a lot greener. There's a lot more mass of plant. That treatment is really important, right? But what I lay awake worrying about at night is that relevant? Does that scale up, right? So I need to do my experiments at the scale of the organisms, because if I'm thinking that that change in the environment is changing how individuals interact, compete for nutrients, for example, I need to do it at the scale of the organisms. But oh my God, what if it doesn't matter? What if it's not relevant to some other place on Earth? And so here's my quadrat. This is the standard metric of all grassland ecology, 1 square meter quadrat. That's what we sample. But then I take that and I say, okay, I want to make some inference about the field. And I'm like, okay, I can probably do that. I feel pretty confident I can infer something about what's going to happen in this field. Then I'm like, okay, maybe like all of Cedar Creek, which is a pretty big place. So I'd feel okay, well, maybe not in the forested parts, right? But can I say anything if someone from downtown St. Paul who's at the Capitol says, oh, tell me what's going to happen in future Minnesota? I'm like, I don't know, right? Or what about this challenge that we have as ecologists? We want to be able to say something at the scale of a region, at the scale of a continent, or, it'd be nice to be able to say something about the world. But we're constrained, right? I'm sampling in 1 meter square plots. I'm applying my treatments at 5 square meter areas, and I'm not even looking in other ecoregions. What can I know? What can I say? So here we are, and I just want to really highlight this problem. So if I'm interested, for example, in nitrogen and grazing rates, and I'm sampling at one point, so that's Cedar Creek LTER, it is one combination of land use or grazing and one combination of the rate at which nitrogen is coming into that ecosystem. But what I don't know is, what if the plants of a different evolutionary origin were, experiencing these treatments? Would they respond the same way? What if I wanted to make some projection about what's going to happen in India with an entirely different combination of grazing and nutrients, or Australia with a fundamentally different, set of conditions under which these treatments would be applied, or conditions under which that future will occur? Okay, so what I really need to do is to replicate my experiment under all these different conditions, because that would be really cool, and that would then allow us to understand whether, grasslands around the world are doing the same thing. But that is logistically insane. Like, I can't put in, 50 sites around the world and do the same experiment. But that's what we're doing. So this is the nutrient network, and I'm not going to talk about the genesis during my talk and sort of how we made this happen, but I'm happy to answer questions about that later. This is a bottom-up collaboration. A handful of us decided that these were the questions we really wanted to address. We designed an experiment, and we wrote letters, and a lot of people said, wow, that sounds cool. I want to do it too. And so it's these points around here, these are all the yes people in the world. They're like, yeah, I don't really have time, but yeah, I'll do that too, right? I'll shove it into my not time. So in case you're wondering where they are, they're right here, at least the ones that work in grasslands. What we're doing is a replicated experiment under all of these different conditions: evolutionary origins, rates of nitrogen deposition, any number of types of herbivores. And these are some of the people involved, and yes, That's an elephant in my plot. I've had people ask me that. Okay, so what does that look like? What are we doing? At each site, we have 3 blocks per site, so 3 replicated sets of plots. This is being replicated in more than 100 countries, more than 100 sites in 24 countries around the world. And I don't have any money for people. People are opting in to do this. So this is voluntary. It's not like I could set out a grid and say we need a site here. We have an observational data set across all of these sites. Then a multi-nutrient, so a full factorial combination of exactly the same rate and type of fertilizer being put onto plots, nitrogen, phosphorus, and then potassium and micronutrients as one. So that's for 2 to 3 for 8 plots. And then we have this top-down, bottom-up, or fence by fertilization experiment, where we take advantage of the all-nutrient and no-nutrient plots, and we put up a fence around that same treatment. So we have really a second full experiment that we can look at to look at interactions between herbivores and nutrients. So in each of these plots, the core response variables that we're measuring, this is not citizen science, this is being done by experts, so people that really know their botany and experimental design. Species cover, so the percent cover in each of those 1 meter square plots. The live plant mass and litter, we clip it, dry it, weigh it, and separate it into groups. Separate it into groups and then weigh it. And then litter, so dead mass, last year's growth. Light interception above and below the canopy, so we can look at another axis of resources. Plant provenance, so at the scale of a site, we know whether a species is from there or not. And there's all kinds of cool parts of that piece of what we're doing, which set it apart from some of the species invasions work that's been done before. Happy to talk about that. And then soil texture and chemistry in year 0, or that observational year, and then years 3, 6, 9, because it's expensive. And it also, we don't want to have Swiss cheese plots in a long-term experiment. And then at every site around the world, we have between 1 and 10 years of data at this point, which is pretty amazing. So we have about 40 sites where we have a decade of data that are spread across 5 continents. So what does that look like in words? Currently we have 108 sites, but for example, a guy in China just wrote me and said they're putting in 10 sites this year. They'll put in 10 to 20 more in a big transect across China next year. So that's whatever. 10 to 30% more sites. NorthNet is now going in, so a sub-network doing exactly the same experiment, but just in the northern latitudes. So Finland, Iceland, Sweden, Canada, and then in Alaska are going in this year. So it's growing, pretty rapid clip. It runs from Argentina, so Tierra del Fuego, up to Svalbard, Norway. So we've captured pretty much grassland extent, north-south. We have about 3,000 plots that we have data from across those years, and about 2,500 plant taxa, which is exciting because it's now grown to be more than 1% of Earth's non-tree vascular plant flora, which I think is pretty cool. So we have very wide phylogenetic representation as well. Okay, so that's a lot of data. There's a ton to deal with. What are we doing with that, right? There's a lot of different methods to tackle these data and to address the questions that I raised at the very beginning, these multifactorial questions. What do you do with that? So, for example, we have really global representation across, mean biomass running from almost nothing in some of the desert grasslands up to grasslands that are very difficult for me to walk through. It's this much mass of big hefty plants, so 1,500 grams per meter squared. It's pretty insane. Mean species richness in some plots is 1. So, for example, salt marshes all the way up to the Tibetan Plateau has something like 45 species in 1 square meter. That's crazy. And elevation, right? So running all the way up to the Himalayas and up in Colorado, and mean annual precip going from almost nothing, 250 250 millimeters up to more than 3,000 millimeters a year. So we're capturing really, really big gradients, which is cool because we might be able to find a signal in that because our axes are big enough. But how do we ask those questions? How do we inform that? So I'm going to start with the simplest theory you've ever run across, right? Because that is a great starting point. So I make this argument to my undergrad ecology class that you can learn a lot from very, very simple models. They make really interesting predictions, right? So I should walk the walk. So I'm going to walk the walk here. And let's see how far we get with even just the simplest possible representation. So let's think about productivity or plant mass at the end of a season, some aggregate measure of how much plant grew there and net. All right, so let's start with little r, which is births minus deaths at a per capita level, right? So individual net rate of increase. All I'm going to do is I'm going to give you a logistic equation here, right? You know what that looks like. It looks like that, right? So if every individual at a super simple level, right? Let's say every individual has the same mass, okay? If I blur my eyes, maybe that's about true, like compared to trees and grass, like stuff in grasslands are all, let's say they're the same mass, right? So then we can come up with some mass here, total mass, right? We're switching units. That's pretty straightforward. Okay, so that leads to a prediction, which is pretty cool. If I increase K, right? Maybe I add nitrogen, and then I add phosphorus, and then I add potassium and micronutrients. Maybe that's just simply increasing K, right? Super simple. But what that suggests is that I should just see total increase in biomass with number of nutrients, right? As I add these nutrients. All right, let's start there. That's good. Now I'm going to move over to this grazing and fertilization. That is about the simplest possible representation of grazing, right? Some linear offtake rate. I've got my herbivore and I've got my plant, right? It's getting eaten. And then, I've got herbivores growing and stuff. But we're going to ignore this, right? So this alone gives us a prediction, right? Very simple prediction. Dumb maybe, but herbivores are predicted to reduce mass. All right, so I'm going to just stand to the side here for a sec and say, this is really useful in part where it's wrong, right? Because I can start with this, and if that's sufficient, awesome, right? Stop there, that's good enough. But if it's not, if it doesn't map onto this, then maybe I need to start adding in some more biology. But I might be able to stop with this. So let's see. We can test it. Oh wait, I had this great idea. Why don't we go out in the field and add nutrients and put up fences, right? And so we can test this across a lot of different sites because the math theory tells me that the same thing should happen everywhere. It should always be the same. There is no contingency. Okay. So as an empiricist, I'm like, yeah, that's nice. Let's see. Let's see when that works and when it doesn't. All right, now let's switch over to diversity. This is where it gets, now we're to the fourth lecture, maybe, in my undergrad ecology class, so hold your hats on here. Grazers and fertilization, nutrient co-limitation, start to think about trade-offs, right? So we might have some trade-off between species. So start here with a Minot model, right? So we just have resource uptake here, right? So we're to like the third or fourth lecture here. And what this tells us is that for a K within the Minot equation, I'm not walking through them. Should I walk through the Minot equation? Probably not. Right, so for the K, it gives us this prediction that this population or species with the lower K, right? So this more rapidly growing species should be the one that wins, okay? So that's great. Now we just put in some density-independent mortality. Again, the dumbest possible description of mortality, right? And then we can come up with an equilibrium, right? So we've got the growth rate and we've got the death rate, right? So we can look at what is the equilibrium for each of those species, the R star, the resource at which births equal deaths for that population, right? That's really cool because it then gives us this prediction that for two species, the one that replaces itself on the lower resource should win. Okay. That's kind of neat. Now let's extend that, and it's not worth looking at all the equations, so let's look at it graphically. Whoops. There we go. One species, two resources. You guys have seen this for the most part, probably. Okay. So if I have two resources for this one species, it requires this amount of resource 2. It requires this amount of resource 1. With that, it then has positive population growth rate. Without it, it declines to extinction. Okay, that's pretty straightforward. Now, we've got this ratio of resources that are implied here. So the resource uptake rates. And from that, if you took an undergrad ecology class, you'll remember, right, that you can then define a zone of coexistence by looking at the apex of these two species, where they cross, and looking at the resource uptake rates. Within this range, both species have positive population growth rates, so that resource supply rate. All right, that's cool. Now, I'm going to extend it graphically to a whole ton of species, some of which would or wouldn't persist in that community, but I don't really care. I just drew a bunch of cool, pretty lines to show you generally what this would look like. All right, so now let's take that extended one for a community. That's a community of plants here. 2 resources, lots of different species that have different resource requirements on which they can have positive population growth rates. Now, if I have this rate of nutrient supply, so this is getting closer to what I might be talking about and able to test, right? A low rate of nutrient supply. At an intermediate rate of resource supply, I might, for example, find more species coexisting. And then when I add further resources, I might find a decline again. Now this gives me some really cool, really simple predictions to test. First one, if I'm in a diverse community, let's say this one, right? Adding resources should lead to a decline in the number of species present. Okay, it's pretty straightforward. If I add 2 resources, for example, at the same time, I might maintain coexistence, right? I might not. It depends relatively where that falls in these zones of coexistence. Okay, so that's a prediction about diversity. But the other thing that you can see here is that each of these colors represents a different species, right? And so you see that we have a change in what species are coexisting as you move along this resource gradient. So we should see turnover of the species present in the community along a resource axis. So I can test both of those using grassland compositional data. I think I just said that, right? Adding nitrogen or phosphorus, for example. All right, so what's really cool is that by replicating this empirical work across a whole bunch of different background environments, I can then move beyond demonstrating, yeah, this theory can work, right? So at Cedar Creek, this work has been demonstrated. It works at a first approximation, right, to describe what's happening in the field at Cedar Creek, right? But do we know whether it's relevant around the world? No, we don't. Right? And so this allows us to then say, under what conditions does this really work? Does this actually play out everywhere? Or is it only sometimes? When? All right, so moving on to actual data. So if I have my 2 axes as nitrogen and phosphorus, or phosphorus and potassium, or I extend this out to be a 3-dimensional description of what's happening, right? I can make these same predictions about coexistence, but I can also test those predictions from a few minutes ago about productivity. So do they play out? Well, yeah, kind of shockingly. So this is on the x-axis is change in biomass. On the y-axis is change in the number of species present in a plot, so coexisting at the local scale. And what these numbers 1, 2, and 3 represent are the number of nutrients. So not the identity, but just simply how many did we add? Did we take it this direction? Did we take it that direction? Did we take it out on the diagonal in that set of axes that I was just showing you, right? And so what you can see is that with increasing numbers of nutrients, just adding more, you get more biomass. It doesn't matter if it was nitrogen or phosphorus or potassium. When you add all of them together, you get a greater increase in biomass than you do if you add nitrogen and phosphorus, or phosphorus and potassium, for example. You also see a greater loss in species diversity with increasing numbers of nutrients. Not necessarily identity. In grasslands, partly because a lot of work has been done at Cedar Creek, which is a sand plain and has no nutrients in the soils. Much of what we assume to be true about grasslands around the world is that it's nitrogen. It's a nitrogen story. Forget about phosphorus. In fact, it's been almost shocking to some people that I'm sampling phosphorus at Cedar Creek. Why would you do that? But it turns out it matters, and potassium actually matters at Cedar Creek. But these are global means. This is across dozens and dozens and dozens of sites. I think there's 54 sites that we were able to include in this analysis. I can't remember the total number. Okay, another prediction is identity, right? So the number of added resources, we expect that with increasing numbers of added resources, we would probably move away from the composition. Like we would move further and further from the original composition. So dissimilarity. These stars represent dissimilarity among plots just before adding any nutrients. So these were the plots where we subsequently added 1, 2, or 3, but the pre-treatment difference in community between control plots and treatment plots. This is sort of the ambient dissimilarity, if you'll have it, among plots across grasslands. But you can see that adding 1, 2, or 3 nutrients increases the dissimilarity of plots in their composition from the original set of, or the control plot, sorry, not the original. And this is taking into account both space and time control. So how different are you from your original, and how different are you from a spatial control? So that's cool. That also bears out. It says, yeah, you know, at a first approximation, that super simple theory from like the 3rd week of your undergrad ecology class actually kind of works, which I have to say I didn't really expect. But there are some really neat things that we've also learned by doing this through time, for example, is all of that theory that I just showed you, that's all equilibrium theory. Like, we're assuming that everything came to an equilibrium like that, and we're just going from there. This is not an equilibrium. So what I have in color from blue to red here is time. So number of years since the treatments were initiated. So year 0 is pre-treatment. And what you can see is that the number of added resources and diversity decline. So you have a greater decline after more years. So there's clearly, we're not in an equilibrium, right? You're seeing this greater effect of 3 nutrients even in early years, but it's an increasing effect through time. So we still need to keep looking. There may be something that, you know, maybe it'll stop. Maybe we'll stop losing. When will we stop losing species? That's a really important question to know. All right. Diversity loss is greater at sites with a larger species pool. So this is the total number of species. And you can see that this is comparing 0 to 3 nutrients added. So I don't have all of them here. But from 0 to 3, you see that when you add all 3 nutrient types, where there's not a lot of species to lose, we don't see as much loss. Where there's a greater gamma pool, total pool of species at that site, you see more species are lost. Again, this would be really interesting to plot out through time, and we haven't taken that out, projected that out through time to really look at this effect. But clearly there's a contingency here that matters. We need to include that piece of biology to make predictions across these grassland sites. Okay, now I'm going to shift for a moment to think about diversity and productivity with herbivores, because I think herbivores are great, and I want to talk about those data because they're really neat. This framework is a problem, though. Like, I can't just fall back to this when I think about an herbivore, because these are about resources being added, right? So if you go back to the Manoa equation, right, that's about resource uptake. It doesn't do a good job capturing what herbivores do, right? They're eating, they're consuming that mass. But what if we think about an herbivore in a slightly different way, as maybe increasing light at ground level, right? So they may be removing mass, but they may also be giving species access to a resource. So what if you add nutrients and you get more biomass? Maybe there's more light that's captured, but maybe the herbivores are removing that and relieving that light competition. Okay? So you can imagine that then could map on as another resource, leading us to a lot of this or analogous predictions. All right. So let's think about the impact on plant productivity. So generally in grasslands, unless you work in Central Africa, the dogma is that herbivores don't really have much of an impact on grassland mass. If you work in Central Africa, they do. But these literatures almost don't speak to one another. It's very interesting across the cultural, anyway, I won't even go there. The cultural part of the science is really interesting. So what this shows is fencing, nutrient, and then the interaction, nutrient by fencing. The nutrient I've just shown you with the Harpole et al. Work, led by Stan Harpole. This is short-term and long-term, again, because I've become really interested in these time scales, right? Because it looks like maybe there isn't an equilibrium, or maybe there are some cases under which we do reach an equilibrium pretty quickly, right? And I want to start to understand that more, because we're sort of falling back to this fairly simple equilibrium theory as a starting point. We get a pretty similar answer with nutrient addition in short and long time frames. But herbivores, that one kind of blew my mind. I mean, we're looking at kangaroos that are really thought not to do that much to grasslands, and deer, and like they don't even eat grass, right? And rabbits. Has anyone been to Silwood Park? No. Silwood Park in England, like all the grass is about this long. It's totally controlled. The mass of grass there is completely controlled by rabbits, right? So these, what? Getting a rabbit. There you go. Talk to Mick Crawley. So anyway, right, so there's this huge variation in what these herbivores are doing around the world, but on average, which kind of blew my mind, I thought the variance would be way too big to pick up any effect. On average, around the world where we're fencing out these herbivores, we are seeing an impact on increased amount of plant mass. I could talk more about that. I don't think it's just from consumption. But that maps on to my fairly simple world of, herbivores removing some of that plant mass. They're not fully keeping up. There is a developing interaction here through time. All right, but let's think about diversity. So again, this is the all nutrients added fence and then the nutrient by fence interaction. And as someone who thinks that herbivores are really cool, when I first plotted the main effects here, I was a little bit disappointed. I was kind of hoping that herbivores would do something. But what we find is, again, like the third time I've shown you this, that nutrients really impact what's happening in grasslands at this local scale. We see a local loss of diversity. Again, like 2 species per square meter, which is kind of astounding. But overall, we don't really see an effect of herbivores on grassland diversity, and no real interaction between the two. What's really interesting here is that there is a change in identity, though. Right? So we do see that shift in identity of which species are being impacted. So this is work led by Eric Sieblum, Nutrient Fence and Fence by Nutrient, looking at the identity of these species defined through, are they native to this site or non-native to this site? And what we see is that when we add the nutrients, there's a big difference in the response across these two different categories. Differentially, we're losing native species out of these systems across grasslands. Exotic species, yeah, whatever, they don't really care. They're like, great, bring it on. So it's really the native species that are getting hammered. So this is really a function of loss of one group, one category of species. But what's really interesting is when I take that fence effect and I turn that variance on the x-axis, right? So now I have the effective fences on light as a quantitative variable, right? So where removing herbivores reduces light at ground level, so where that happens, so that's a contingency, we see a loss in diversity. So there is that loss of diversity where herbivores are impacting the access to light at ground level. That was really interesting to me because that suggests that, yeah, maybe kangaroos don't really matter, right? But it may be that at these sites where we have certain types of herbivores, or really more about the herbivore-plant interaction, that's where we see the loss of diversity when we lose those native herbivores. So they are rescuing plant diversity or maintaining plant diversity, depending on how you want to think about it, at certain sites. Again, dividing that out, again, led by Eric Seablum, dividing that out into native and non-native, again, it's those native species that are the ones that are being lost. So they're being lost on this axis of resource availability, both nutrients and light. Right? So they're not competing effectively in these novel environments that we're creating on Earth. So we're differentially losing those native species. Okay, so now I want to circle back around and say, oh, but what about herbivores and nutrients, right? So what about, not herbivores, diversity and nutrients. So like this number of added resources, we see, light, that was what I was trying to say. We see a change in light. So adding these resources here, 1, 2, 3, we see a decline in light at ground level. So again, it's really this combination of factors. It's no one single factor that's acting to change diversity or change productivity in these systems. And so we're we're thinking very much about the deeply interconnected pieces through this matrix of nutrients coming into ecosystems, changing biomass, potentially changing diversity as a direct effect or an indirect effect through cover, litter, changes in light that are more indirect impacts on changed diversity. Similarly for herbivory, changing maybe the light environment or where it changes that light environment through changed biomass or cover, then impacting more indirectly diversity. Right? So it's not a single, simple set of relationships. It's not just, add nutrients, lose diversity, right? It's much more complex than that. And theory has been helping. Falling back to the simplest possible theory has helped us to dissect out pieces of this. What would we expect in this relationship or this relationship? And then testing that and asking, under what conditions does that matter? Under what conditions can we then make inference based on that theory? And so what this is allowing us to do is move beyond is it nutrients or is it herbivores, right? Is it nitrogen or is it potassium? And start to ask, how much or when, where? Is it in sites with large mammals that we see these effects? Is it in sites where we have native plant dominating the system that we see these impacts? We can start by replicating this experiment under all of these different conditions. We can start to parse out those contingencies and quantify them and ask about soils or climate or identity of species, functional groups. And I'd also add that coming from Cedar Creek in particular, a theory demonstration has been a really important piece of what the work at Cedar Creek, particularly by Dave Tillman, but Sarah Hobby and Peter Reich has contributed to ecology. Coming from there, I feel like what this project is doing is taking us beyond that demonstration. Yes, there is a predictive capacity of theory, very simple theory for what happens in natural systems, and then asking about the next step. Right? So asking about, is this general? Does it happen in other places under different evolutionary origins of plants or yada, 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 right? All of these different factors I've been talking about. And is it important, right? So we can start to quantify the relative importance of different pathways through these drivers of diversity and drivers of productivity. And so what I would say for you guys, for this particular audience of, the intersection of math and biology, is that what we're doing is we're really trying to draw from expectations generated by fairly simple relationships described in these communities and test them and try to understand more about global change. Can we use this to identify some of maybe the mechanisms that are changing communities? And can we take information at one site and under what conditions, when, is that relevant to information at other sites around the world? So when can I say something at Cedar Creek and feel fairly confident that it will also be relevant in Central Australia? I want to thank, in particular, Eric Lind, who has just left working with me, but we worked together for 7 years, who's a postdoc research assistant in my lab, who was the main data manager in this project. Eric Seablum, who is a co-lead of this project with me. This is just one example of 1 year's meeting that has something like people from 7 or 8 countries here on 4 or 5 continents that we bring together every year to decide what are the next questions we really need to tackle. What is that group passionate about that we really need to dig into next? And then, of course, the probably 200 researchers around the world that are contributing to this project with their own dime or whatever their dime analog is. National Science Foundation that gave me 1 grant to start this project. And then the Institute on the Environment, which is a cross-cutting institute within the University of Minnesota that has seen the potential of this project and helped us with some bridge funds to maintain the project through time. Cedar Creek has been amazing, and we have managed to keep some funding for the postdoc position through that. So yeah, of course, University of Minnesota that, you know, pays me, which I also appreciate. So yeah, thanks you guys. Because I might not be able to hear you. I know.
Hello. I have actually two questions. In your analysis, I was wondering how you sort of average those data and plotted these confidence intervals, whether you use kind of generalized linear models or generalized additive models kind of thing.
Sorry, what was the second option of models?
So I was wondering, like,
Yep,
how do you
generalized linear
average?
models or-
Yeah.
Yeah.
Whether, because they are coming from different sites. So I was wondering whether they are, or otherwise,
Yeah,
whether you
so
pool them. Like,
there are 200 scientists across the network. So as you know, scientists have some philosophical differences in how you might deal with various different data. There are some analyses that have been done using more of a Bayesian approach. Some of the analyses have been done taking a more, you know, sort of GLM type approach with, you know, nested, obviously accounting for the design, right? So the nestedness of site and plot and block, or block and plot. Right, and so, yeah, I'm sorry.
so my second question is,
Yeah,
my second question is, so there's like, when you're taking data from across the world, there's like, the like kind of factors like temperature
Yeah.
has a like a very systematic gradient.
yeah.
And I was wondering whether you had to deal with those also.
Yeah, deal with. Actually, we embrace it. It's, I mean, deal with sounds like it's a bad thing. I actually think it's wonderful. So the fact that we have these incredibly wide gradients, we've used those in analyses as covariates to try to understand, you know, is it under, for example, highly seasonal sites where we see whatever, greater loss of diversity. I mean, any one of these responses that we're looking at. So we have modeled nitrogen deposition data that we are using. We're using modeled temperature for most of our data. And the reason we do that is while there's really good coverage across the US and across Europe in measuring temperature, even temperature or precipitation, there are other places in the world where it's almost impossible to get really good, especially precip, but climate data. So what we've opted for, unless the questions really require a local, directly measured variable, is the modeled data so that we have more consistency. So really, across the network, we've tried to make choices where we're going for consistency. So all of our methods and the data that we use. Yeah.
About the network issue, of course, there's tremendous problems with scaling up and tremendous problems with not sampling all of the variety of things going on across the planet. But you have an ideal circumstance, and I wonder if you've done it, to basically subsample from a collection of sites and see if the implications from that subsample are also justified based upon other ones. And how large a subsample does it take to actually say we have robust conclusions?
I love that, and it's something that, honestly, I thought that one of the early career people in the program, or in the project, was going to do for their ESA talk this year. So I intentionally stayed away from that. But I love the idea because we could basically say, for example, we have a 2011 paper Peter Adler led. In that paper, we had, I don't remember, 30 some odd sites, right? That was the size of the network at that point. What we could do, albeit I kind of want to do it behind closed doors, is we could take that subset of the data out and use all of the data since then that have been collected at whatever it is, 70 some odd sites, and we could say, did we get the same answer? Right? Because that first set of sites was not spatially random, right? It was who I knew, right? And who a few of us knew. So it was mostly North America, Europe, and Australia, right? So we have much broader spatial representation at this point. So I think that's a huge question in ecology right now, is like, well, could we do it again? Do we get the same answer? And we do. We have that opportunity, and I haven't done it yet. But now that I know that person did not present it at ESA.
I might have missed it, sorry.
No, I'm saying she didn't. No, we talked later and she's like, I actually felt uncomfortable doing that as an early career person. I didn't want to put myself out there. So because the Adler et al. Paper is, whatever, I don't need to go there, but it's had
could be
more
wrong,
of a
right?
barbed, well, no, it's just had more of a barbed reception than we ever expected.
Hi, thank you very much for your presentation. That blew my mind. So
That's my goal.
So I have two questions. One is, you saw through your data that increasing the amount of resource, nitrogen for instance, you decrease biodiversity. And you showed before there is some prediction of increasing nitrogen for 2050. So do you expect like we lost a lot of biodiversity due to that?
Sorry, tell me the second half, the grazing part.
So the second part is like you show some projections that in the future is going to increase the amount of nitrogen.
I see what you're saying. Yeah. So do
So-
we expect to see a loss in diversity? Yeah. So we know it's happening. Actually, not as clearly with the diversity, but I can tell you about productivity clearly. Eric Lind and Carly Stevens, who's in the UK, got together and led a paper that came out a couple of years ago where we'd never really looked across nitrogen deposition gradients and had exactly the same data to compare. They did that analysis and found that there is a distinct signal on top of others that you'd expect, like where it rains more plants grow, right? That's not super surprising, but where there's more nitrogen deposition separate from that, we see greater biomass. And so if that biomass is depleting light at ground level, we would expect that we would then see loss, particularly, I'm guessing, of short-statured natives. So we haven't gone there yet, and the data are not as clean, and I suspect that that's because herbivory is very different among sites, so we haven't yet extracted that. Yeah,
My
but it's a good
second question is about how is the dynamics of working with so many people?
Oh, it's a great question. It's like the most fun science overnight camp I've ever had. I can make up just so stories in retrospect about why the network is as awesome as it is, but I suspect that there were a bunch of pieces that came together to make this the most fun group ever to work with. One is we have no money, so no one can come and ask me for money. Everybody is self-funding these experiments. We kept them inexpensive, and we kept them relatively low in terms of time consumption and personnel consumption. And the unintended impact of trying to fit something into my own overfull life and designing it that way is that it's meant that we've been able to have collaborators in areas where science is not as well funded. And so it means that we have early career scientists. There was a grad student who was going home to South Africa. He was like, hey, I want to put in a site at home. You know, I'll just do it. It's out back on my parents' place, right? And there's this guy who's an aquatic ecologist in Europe. And he's like, hey, I'll just do it in my parents' backyard, you know? Because they were really engaged with the questions, right? And so it's people that are passionate. They're doing it for no money, so it's really passionate. And they're all the yes people. They're like, yeah, my life is totally full. Yeah, I'll do that. You know, like I'll fit it in because it's cool. And so I think that because of that, it's been this hard filtering where we just end up with the best people ever. And it's really creative and exciting. And I think I also embrace difference. I like it because it makes the science more rich. And I think it makes us, you know, I think that's why we've had as many, you know, heavy-hitting papers as we have out of the network, is not because any one person is brilliant, although everybody, you know, there's a lot of really smart people in this network, but because the internal peer review is like the hardest thing I've ever gone through. You get comments from 60 people on your paper, right? You know, like I just sent out a paper to the network yesterday. I've already got like extensive comments from people on, you know, you haven't thought about this theory, but I rewrote this section. I mean, it's like awesome. So I've found it really fun. We've set some internal rules, and I think that's helped because people know what they're putting in and they know what they're getting out of it. And we've tried to make the, you know, cost benefit be appropriate for all of our academic careers. So I don't know. I think those are pieces, but I'm not really sure. The pieces come together in a really great way of people around the world that are just super fun. So I host a dinner at my house every year. I just make dinner for everybody that comes to the meeting because I just feel so thankful that all these incredible people are willing to contribute to this project. So I'm like, hey, you're in Minnesota. Let's have corn and pesto. It's August. So yeah. So I don't really know, but it's a fun ride. It's really fun science.
I have a question with regards to herbivory and the interaction of exotic and native species. Diversity reduction under nutrient,
Yeah.
more nutrients. So have you considered that the herbivory that you're excluding is only limited to large animals, so you are not excluding, for instance, insects?
Absolutely.
And that could have a negative effect mostly on natives and not on the exotic species. So do you have any comments on that?
So you're saying that by only excluding the 50-gram and larger mammals, we would have that they would be the ones. Sorry, tell me. I'm missing the hypothesis about the native. So the native larger or the larger herbivores,
right? So
are rescuing that diversity.
So insects would be feeding on only the native plants under nutrient-rich conditions because they are nutrient-rich, right? So you have larger plants growing more profusely that would be infested by native insects. And they obviously can't feed on the exotic plants. So
Yeah, I don't know. I mean, you've got a couple nested hypotheses
Right.
there, right? So there's an interaction implied in what you're asking, but also, so here's my simplistic, what I can say from my data kind of answer, which is insects can get anywhere, right? And I'm fertilizing inside of fences and outside of fences, and we're not seeing a big effect along that axis. It's really, is there a fence there, and is that reducing the amount of light at ground level? Where that's the case, then that's where we're seeing the loss of native diversity, but it's very different inside and outside of fences. So I guess I don't see a lot of support for the insect hypothesis, because then you would expect fertilized, not fertilized, from what you were saying, right? So when you're fertilizing, you would expect the insects, right? So then I would expect a much bigger difference for the natives across the fertility gradient than across the fencing gradient. I don't know if that's right, but at least from our data, I don't see evidence of that. Another piece of that is we have at a subset, like 25 sites or so, we did insect sweep sampling. So we've got all kinds of additional data. So I talked about the core data, but we have, nitrogen mineralization and insects and, all kinds of stuff that we've done. So with that, we didn't find a huge impact in terms of biomass or diversity across those treatments. But we did find sort of I guess the mass changed across the nutrient addition, but not the impact in terms of the natives and non-natives. So, I don't know. I think it's a nested issue, though.
I'll have to think about that. I'm not as familiar with your data.
Yeah. No, I'd love to hear your thoughts on it.
All right, one last question from Dan.
In some of the NewtNet papers, I can see that what are exotic species in one place are native in others. And
Yeah.
and you have the data.
Yeah.
And it makes me wonder, would it not be more interesting than looking at exotic versus native response to various things, to look at some aspect of function or biology of the plant or
Yeah.
systematics as a proxy for it? Or have you, and I haven't seen those particular
So yeah,
papers.
there's a lot of papers that have come out of this project. Jennifer Fern, who's at Queensland University of Technology, and is just like smart and wonderful. She has led a few papers where she's taken that subset of species that we have within our data set that are present in both their native and non-native ranges and looked at their responses to ask, are they doing something different? In some cases, yes, mostly no. Mostly we could predict what they're going to do in their non-native range from their native range. There are a few cases where that's not true, but really across the board. Yeah, so that's the thumbnail. Yeah, but it's a really neat opportunity to look at that. Jen is leading a second paper now looking at the species responses to the nutrient treatments and the herbivory treatments. So the first one was just looking at change across the observed gradients. So, yeah.